Howdy. So today I'm going to talk about what we learned this past week in my biology class, and that was a pretty information-heavy set of chapters. We talked about the nucleus, and we also talked about DNA transcription. And to start off, we as humans are eukaryotes, and that means that inside every single one of our cells we have a nucleus, and when you look at a microscope you can actually see it's a pretty defined little area, and that area is really important because it contains all of our genetic information, which is packaged as chromosomes. And chromosomes are really tightly wound packets of DNA, little strands of double-stranded strands of DNA, and they're wrapped around these little globular proteins that are called histones. And if you were to take those chromosomes and unwind them off of the histones, you would get strands of DNA that were almost 6 feet long for each nucleus, which is for each chromosome, which is pretty impressive. So anyway, that DNA is all coiled up, but we want to be able to use it. And because DNA can't travel outside of the nucleus, or it shouldn't, we have what's called RNA, which is complementary to DNA. And RNA comes in 3 forms: messenger RNA, transfer RNA, and ribosomal RNA. And there's some other ones, but those are the 3 main categories. And the importance of RNA is that it's able to leave the nucleus and go carry out a bunch of different functions based on the orders that it's written on the little code. So the first thing we would do when we have a code, right, is we want to transcribe it. So the first process we have is transcription. That's what we talked about this week. And transcription begins when the DNA unwinds, and we have transcription factors and activation transcription factors that will bind an enhancer and basically tell RNA polymerase, which is what's going to make our strand of pre-RNA, our transcription factors and activator factors, that's going to tell it where to bind and where to start transcribing. And just as a side note, one really important thing about activator transcription factors is that there's one called P53, which is really important for a lot of different types of cancers. I actually told you about in a class 2 semesters ago when I talked about skin cancer, and that actually plays a really big role in the development of skin cancer because P53 is able to suppress tumors. So when there's errors there, that's how we get cancer in a lot of cases. But anyway, so our DNA is unzipped, and we'll have what's called a coding strand and a template strand. And our transcription factors are going to tell this RNA polymerase where to bind, and it's going to move across the template strand. And so it is going to be basically a mirror image. We've got 4 sets of nucleotides that are matched up, and our RNA polymerase is going to move this direction, and it's going to crank out pre-RNA from the 5' to the 3' direction. And so what we're going to have then, once that process happens, is a strand of pre-mRNA, typically if this was mRNA, and this strand is going to have a cap. It's going to have some exons and some introns, and then it's going to have a tail. And the cap and the tail are just sort of so we know where to stop, where to start. And these exons are just are what we're going to use, and they're going to be extrons, express, and the introns are going to be taken out by these things called spliceosomes. So I don't know if you can see, but I'm erasing them. My finger's the spliceosome. And so we're going to be just left with our exons. Very hard to say sometimes. And this strand, in this case, is going to be mRNA. And this can then take a message from the nucleus and leave to other parts of the cell to give messages, to synthesize different things, and we're going to learn a lot more about that next week. So I really hope you were able to follow this video, but that is kind of the gist of transcription and the nucleus, and I will be back in a couple weeks, hopefully, to tell you more about what I'm learning in class. So thank you for bearing with me through my first video and the fact that I could not figure out how to flip the mirror. Bye, Grandma.